So I'd like to thank the organizers, of course, for allowing me this opportunity to speak. And while I was getting this talk together, I began to muse on the fact that in somewhere late 2006, 2007, not 10 years ago, PNAS asked me to review a paper by somebody named Sebastian Fraun, and the other person was Thomas Bosch. And I read the paper, and I was absolutely blown away, absolutely blown away. So I wrote my review, and from a symbiosis person's standpoint, this was clearly a benchmark paper. It was the first paper out of this particular effort. And I got a call from the editor saying, "Why do you think this is a benchmark paper?" So I spent some time talking with the editors, and I said, really honestly, in 10 years, you're going to see that this was a really benchmark, this was a very important paper. So as I was getting ready for this, I thought, my God, it's only been 10 years, and look at what has been put together here. I mean, it is magnificent, and I just want to congratulate Thomas on such a meteoric rise in this field. So what I'm going to tell you about today is I'm going to tell you about some work that I do on this, the squid-vibrio symbiosis. And I just recently moved back to Hawaii, and I'm very happy. This is the lab. This is where my lab is, Kiel Marine Lab at the University of Hawaii, in the School of Ocean and Earth and Space Technology. And here I am at night collecting animals in the field. And I love being in the field, so I'm very happy. So I will be telling you a little bit about the system, and I'm going to, focus on one thing. I should tell you that I had, about a month ago, a very severe respiratory infection, and so I'm hoping that I make it through this talk. This is the star of the show, Euprymna scolopes, and it's a small night-active predator in the Hawaiian archipelago. And if you were to do a ventral dissection, I want to apologize for all of you who've heard this 100 times, but if you do a ventral dissection, you open it up and you see this conspicuous bilobed light organ in the center of the mantle cavity. And if you do a section on that, you see that there's a series of tissues. In the very middle is an epithelium that houses Vibrio fischeri, the luminous bacterial symbiont. It houses Vibrio fischeri extracellularly along the apical surfaces of epithelia. Surrounding that set of epithelia is a set of complex tissues that modulate the light, control the amount of light that goes out into the environment. And it's amazing, it's just like a backwards eye. And in every way, all the way from the morphology down to the gene regulation. It's an amazing system. So the bacteria, Vibrio fischeri, for different portions of its interaction with the host, interacts with mucociliary membranes as well as eventually and permanently the microvillous surface of the crypt epithelia. And so I'm just showing these here because these are very highly conserved types of cells across the animal kingdom. So in form and function, these epithelial membranes are highly conserved. So this is a vertebrate, and these are the tissues of the squid. So the goal of studying these things in a simple system is we want to define the rules underlying the mechanisms of symbiosis. And by using a simple system, we hope to get down to those mechanisms. And you want it something that's experimentally tractable, and that's what this system is. And so we've been lucky, in addition to studying evolution and ecology of the association, we've been able to gain insight into the establishment of symbioses, their development, and their stability. And this is a horizontally transmitted symbiosis, and that is it's acquired anew each generation, probably the most common type of symbiosis in nature. I'm going to spend most of my time talking to you today about establishment, and that's because we've been focusing on that in the last few years. In the very beginning, we did a lot on development, but now we've been studying establishment. So in a horizontally transmitted symbiosis, asks the question of how is the symbiont selected against a background of other environmental bacteria? Now, you'll find that I'm very host-centric. This is certainly a dialogue between the host and the symbiont, and it's not just the host selecting, it's the bacteria selecting, but I'm kind of an animal chauvinist, so I'm sorry about that. And so why use the squid-vibrio system for this particular question? Well, the size of the juvenile squid is only a couple of millimeters, and the light organ is about 400 microns across. And what this allows us to do is to put it under a confocal microscope of one sort or another. The timing of colonization also is extremely short, and we've characterized it, and it's got a very characteristic tempo to it. And we've defined that tempo, and so we can study each one of those stages. And so the entire natural process can be studied in real time under a confocal microscope. And so we've been working on this for a while, and I'm hoping that we're getting somewhere. So for those of you who don't know the system, I want to mention that in embryogenesis produces a light organ morphology that's very different from the adult morphology. And the most important thing is that on each lateral face of the light organ is a ciliated microvillus epithelium that is associated with the colonization event. What happens, here's a little baby squid, and they're the size of a Drosophila, as I mentioned, and when the minute they hatch, they begin to ventilate seawater through their mantle cavity, and most of the seawater, the light organ is invested in the funnel, and so most of the water goes by, and this is where capture happens. So the symbionts, if you look here at these three pores on each side of the light organ, each pore leads to an independent duct, antechamber, and crypt. And so there are 6 independent chambers that house Vibrio fischeri, and those 6 chambers remain throughout life. They elaborate, but they're always 6 different chambers. If you did a histological section at the level of one pore, you'd see that the bacteria have a long trajectory that they have to go through. So they go from the pore up this duct and through into an antechamber, which you can't see here, but they go through a bottleneck, and then they come into the crypt spaces, where they reside throughout the life of the animal along the apical surfaces of epithelia, interacting with mollusk hemocytes that seem to sample the crypt space and tell the rest of the body what's going on there. So the colonizing symbionts reside extracellularly, as it says there, deep in the tissues. And so what we have spent a lot of time doing in the many years that we've been studying this symbiosis is to try to understand development. And what happens during development is that the bacteria induce the loss of the tissues that are associated with colonization. And so one of the major questions is, how does that message get out to the surface? And so, but I won't be talking about that today, but I just wanted to show you how deep inside the tissues those bacteria occur. So here's the journey that these bacteria have to undergo. So the symbiont cells pass through 5 territories. And these are 5 very biochemically different territories. So imagine a human taking in a symbiont, like Helicobacter pylori. And in this case, what happens is they go across 100 microns. When a baby takes in, say, Helicobacter, it has to go, many, many, many centimeters until it gets to the antrum of the stomach. How many environments does it go through? And what is the effect of having to go through a series of environments have on the eventual behavior of that bacteria and its association with host tissues? So we're trying to figure out if these environments are meaningful to this process. So the symbiont cells pass through these 5 territories over 100 microns, so over about 50 body lengths of the bacteria. So there's surface pores into the duct, there's an antechamber, a bottleneck, and then they go into the crypts, and after about 12 hours, they're induced by autoinduction to luminesce. So this landscape sets up, as I hope you're convinced after my talk, it sets up a biochemical and biomechanical and biochemical filter that appears to select for Vibrio fischeri. So what is the landscape here? What are we talking about? What is the challenge? So Vibrio fischeri makes up about 0.1% of the bacterial plankton. So, and sometimes less. So about 1,000 cells against a background of 1,000,000 cells, other bacterial cells in the environment. And they are able to resolve within 2 to 3 hours their symbiont. They're able to pull that symbiont out of the water. One of the remarkable things, I think, for the microbiologists is that Vibrio fischeri is in a mixed culture at 10 to the 6 bacteria per mil, which is not very dense. And they go from that and they make a tremendous habitat transition. They go into the animal, and these are RFP labeled and GFP labeled fischeri in the crypts of a juvenile animal, and this is each side of the light organ. And so what's really remarkable about this is they're in there cheek by jowl. If you do a TEM, you see that they're right up against one another. And so when there was discussion of the intracellular bacteria and looking at mechanical stress, I was thinking about these extracellular bacteria and whether or not there's mechanical stress there as well. But so I want to start by talking about what the variation is. And we've Ned Ruby's lab, who's my partner, he's the bacteriology side of this system. He's been studying some aspects of the variation of the symbiont and what is the impact on variation on how they interact with the host. And so we learned very early on, that we're working on this. This is Oahu, for those of you who don't know Hawaii well. This is the island of Oahu. And the lab that I work in is down in here. We collect in 2 sites on Oahu, Mauna Loa Bay and Kaneohe Bay. And we found out that Kaneohe Bay and Mauna Loa Bay have 2 different haplotypes of the squid host. And they're so different that a Drosophila biologist who I worked with, George Roderick, said to me, if they were Drosophila, they'd be different species. So 2 different phylotypes or haplotypes on either side of the island of Oahu. So the question is, do the strains show fidelity to these 2 sides? And Clotilde Bongrand in Ned's lab showed the answer is no. In other words, they're all equally capable. Anything from Kaneohe Bay can colonize just at the same rate as those from the other side. So there wasn't anything there. But what she did find was she found something really fascinating. There were 2 different types of strains. And one type of strain, so in a co-colonization, sub-Vibrio fischeri strains dominate. And some of them are social. And what you mean by that is if you put one of these dominant strains with a strain that she called social, and you put them together and you ask the squid which one they like, they always take in the dominant strain. Whereas if you put 2 social strains together and you ask what goes in, they go in together. And they co-colonize the light organ. And so it was very interesting that there were these 2 very striking phenotypes of the bacteria. What she did then was she was able to give some kind of, she was able to rate each bacterial species. This guy right down here, this social strain, ES114, is the one that all of the genetics on this system has been done by the Ruby lab. And so it's interesting for us now to think about the fact that there are these 2 very different types of strains. So there's a dominant strain and a social type. In Clotilde's studies, very interestingly, she finds that when they're in culture and maybe out in the environment, the social strain dominates the situation. It's a competitive dominant when in culture. Okay, and if you do the same thing in a culture situation. Whereas in Squiddo, the dominant strain dominates. Okay, so that was a very interesting maybe there's 2 different strategies on the part of the species. So then what she did, so with Martin Pultz, we did full genome sequences of, Ned's lab did 20-some species. But this is what happened. You see that they really are two different groups of strains here. And then you could see that a comparison of the genome between the two, there was a 250 kb difference, and there's 250 kb more in the dominant strain. So that was really amazing. It turns out that it's a little enigmatic because it doesn't seem like it's a plasmid situation. It's these genes are scattered around the genome, and how that actually came to be seems to have the microbiologists scratching their heads. And I'm not a microbiologist, so I'm not going to go into that, but I thought it might be interesting to show you guys that. So there's 250 kb, and of course Ned's lab is now in the process of trying to figure out what that 250 kb confers to these bacteria that allows them to be dominant. But my lab is often tasked with looking at the animal. A lot of times the microbiologists, they'll do everything not to look at the animal. And so I have people in my lab who love to look at the animal. And so this is Sabrina Kohler, who joined my lab last year from the Max Planck in Jena. And Clotilde turned to her and she said, could you look to see what's going on with these and where the dominant strain dominates? So where in the journey does that happen? So Sabrina pushed the system and gave them a whole bunch of bacteria so she could look at many animals and look at them very quickly to see if there was a difference. And so if you do this, if you give ES114, that social strain, if you give that to the animal, this is what you get. At 2 hours, you can barely see any aggregation of bacteria on the surface. Here are a few GFP, the social strains. Usually you have a very small aggregate. So at 2 hours, that's what ES114 is doing, or any of the social strains. And this is what a dominant strain is doing at that time. So here's the whole light organ, and you see huge concentrations. So there's some tremendous affinity of these bacteria for this host animal. And this is 2 hours after inoculation into a background of 1 million other bacterial cells. So now we have an idea of the difference and where the dominance is likely to be exerting itself. So I want to go back and talk about, so how does this work? How do they aggregate into these aggregates that are going or would be symbionts? How does this process happen? And how does it become specific? So let's talk about the steps in harvesting. So the first thing that happens is mucus shedding. And the mucus is very special. Actually, the mucus might not be very special, it's just that there's a lot of it. So it's this in blue here, and so you get this huge amount of mucus shedding. And I want to start by talking about the biophysics. So as I mentioned, there's it's biophysical and biochemical determinants. And this is the fruits, I'm working on a collaboration. So I did a sabbatical out at Caltech in Los Angeles. And I began a collaboration with this group. So this is John DeBerry, who's now moved to Stanford, actually. And this is Jana Nauroth at the Wyss Harvard School for Bio-Inspired Design. And Jana Nauroth, actually she comes from Schleswig-Holstein. And Jana is a true genius. I mean, she's just an amazing kid. She's actually the Wyss Fellow. And so she's sort of the driver. Ava's a mathematician who's modeling the system, and Scott Fraser's helping us with the imaging. So some of the things that Jana has been able to do is watch particle capture by this ciliated field, and she's characterizing particle capture. And she's able to show, so one of the things that she showed was that the ratio of particles advected to caught in a system where you're just giving them inanimate, these are not bacteria, these are particles that are labeled, it's 50 to 1. In other words, this system is actually quite poor at pulling in particles. And so there has to be something very specific going on with the bacteria, with the symbionts. One of the things we know, for instance, is that the bacteria attach. They have to attach. So a question that ought to be asked is there are a lot of different particle sizes in the environment. And so Jana asked, is there size selection, biomechanical size selection? So she gave a 50-50 mix of 2-micron particles and 4-micron particles. And 2-micron particles are about the size of Vibrio fischeri. And so what she found was that there is indeed size selection on the part of the ciliated field. And so they are selecting 2-micron particles over 4-micron particles. When you look at the whole thing integrated together, you can really see that. Now, the question is, how unusual is it for the ciliated field of a marine invertebrate to select 2-micron particles? Is that what they do? So ciliated mucus fields are really common in invertebrates. So what is this unusual? So yes, there is size selection, but is it typical? And the answer is no. So a whole bunch of marine invertebrates have been studied for the way their ciliated field works, and at 2 microns, they're typically very poor at retaining particles of this diameter. So this has probably been selected upon very specially to collect particles the size of Vibrio fischeri. Now, the thing is, though, as this would suggest, most of the particles are that size. So something else has to be going on with specificity. But what we're doing here in this is we're looking at near and far field effects of the metacrinal beat of the cilia. And so we have a big program to study this whole thing and the biomechanics of it. And we're doing a lot of mathematical modeling of the situation, sort of iterative modeling and viewing. Just found out 2 days ago that NSF gave us a big grant, excuse me, from the INSPIRE program. So we're very excited. So we've got biophysical and biomechanical determinants. The biophysical are associated with getting the bacteria into the right position. And then there's biochemical. The biochemical seems to be responsible for specificity, priming, and chemotaxis. So you have to winnow to Vibrio fischeri. And this is the challenges, or what has to go on, is you have to get down to a single bacterial type. You have to prepare for the next steps. So what happens is the chemistry of that mucus is a very low set of concentrations of a series of biochemicals that are in very high concentration in the duct. And so what happens is the bacteria sit there, they gather and gather and gather in these aggregates, and they sit there and they change their gene expression in response to low levels of biomolecules on the surface that are antimicrobial, and then they're able to go into the duct. So they get primed at that point, and priming is very important. Now this priming is something that has been very well described in pathogens. So human pathogens undergo this priming. And as far as I know, this is the only place where priming has been described in a symbiosis that's not pathogenic. The other thing, of course, is that the bacteria have to know where to go. They get up in these aggregates, and then they have to have a way to know where to go. So one of the major pieces of the puzzle was put together by this very talented postdoc I had, Natasha Kramer, in collaboration with Phil Rosenthal here. And we really, really appreciated the collaboration with Phil. And what Natasha did was she was working with ES114. And she put the numbers of bacteria in the surrounding seawater that the animal typically sees. That is 1 to 5,000 cells per mil. At 3 hours, the bacteria had 5 bacteria had collected on the surface of the host, just 5, against a background of thousands of others that are going by. And that animal could tell that those 5 bacteria were present. Tremendous resolution. And it changed, that interaction with the host changed host gene expression so that the animal then enriched these antimicrobials into that mucus so that the bacteria would become primed. And so there is a real reciprocal talking to one another that goes on there. But the response of 5 bacterial cells, of the animal to 5 bacterial cells with a robust change in gene expression was very exciting and sort of unexpected. So the specificity, there's this winnowing of Vibrio fischeri, and what goes on is that a set of conditions is created. And just to be specific, there's lysozyme, a peptidoglycan recognition protein. Hemocyanin is broken down by L-cathepsin into a peptide that has prophenyloxidase activity. And there are bactericidal permeability increasing proteins. And all of these are poured into that mucus. The other thing that's really important is that the system goes from being, so the bacteria go from being at the seawater pH of 8 to a pH that's about 5.5 in the duct. So orders of magnitude more hydrogen ion concentration. These bacteria are being exposed to this. And it's absolutely essential that they turn on the genes to acid resistance out while they're in the mucus on the outside. So this priming is they're being prepared for the next steps. So the hemocyanin, nitric oxide synthase, these are things that we know that they're primed to. And then chitinase. The mucus also has chitin in it. And Vibrio fischeri comes and tells the animal to export a chitinase. That chitinase breaks the chitin down into chitobiose. And the chitobiose is in very high concentration in the duct, and it creates a gradient. And Vibrio fischeri must be primed to respond in chemotaxis to chitobiose. So they get primed sitting out there in that mucus, and then they go up that gradient up the duct. So it tells them where to go. So this is what that chemotaxis looks like. I mean, it's very directed chemotaxis. So what is the priming done for Vibrio fischeri? And this is just one example. We've got examples for nitric oxide and whatnot, but I thought this was a fun example. So we're doing a collaboration with Anders Maibaum at the EPFL, who does nanosecondary ion mass spec, and his very talented student, Stephanie Cohen. And so what we've been doing is labeling up Vibrio fischeri with heavy isotopes C13 and N15. And so what you see is the labeled Vibrio fischeri here in the duct. And so look at these other bacteria that are in the duct. It's amazing. I show this to microbiologists and say, oh man, those bacteria look really not healthy. As though, something is going on there. There's some melanization and so on. If you look at this on TEM, it's the exact same image. Here's Vibrio fischeri, and here's Vibrio fischeri, and here are all the other bacteria. So, of course, what we're looking into is the extent to which and why Vibrio fischeri seems to be resistant to whatever these biochemicals are. But obviously, there's a very strong selection that's happening here, and we think it's likely to be prophenyloxidase activity. And they have some kind of a resistance. And why Vibrio fischeri is resistant, we're working on that. But once colonizing, Vibrio fischeri cells are restricted to the deep crypt areas of the organ, except each day at dawn, they expel about 95% of their bacteria into the surrounding seawater, and then they grow up each day. But for the rest of the day, almost all the day, they're restricted to the deep crypts. And how does this happen? So what I mean by there's exclusion from these other portions of the light organ. And they've gone through here. These, like I said, they're on fire with antimicrobials. And so they go through here, but they are restricted. So how does that happen? Well, we're looking at this by hybridization chain reaction. What we want to know is what genes are expressed in these regions, the restricted regions that are unique to those regions. And so, so I thought you guys might be interested in this because this has just been recently developed. And so this is Niles Pierce at Caltech who developed the method, and Scott Fraser who's been tuning it to various situations, mostly zebrafish. So what this allows us to do is fish is not always fish. Type DNA fish that microbiologists use is often, if you want to know the transcription of genes in the bacteria as well as in the host. And this is the way to do it. So we can follow the individual bacterial cells deep into the crypts and find out what they're doing there. And it is powerful enough so that we can localize the expression of multiple host and symbiont transcripts simultaneously. So what this is basically is, I'm not going to go into it in the interest of time, but it's basically PCR within the cell. So you take a transcript that's at very low abundance, and you amplify that transcript to very high levels, and it allows you to visualize what's going on. So you can follow Vibrio fischeri. So these are individual Vibrio fischeri cells. And this happens to be 16S, but we've also done this with the Lux genes, and Ned's done it with a whole bunch of other genes of Vibrio fischeri. So you can watch the genes of the bacteria there. It's really, really beautiful technique. But there seem to be a series of gatekeepers. There seem to be genes that are restricted to these regions. And so what I'm showing here is I'm showing a bactericidal permeability increasing protein, a member of the LBP-BPI antimicrobial. It's an anti-gram negative bacterial protein. And so this is the message for that protein, and it occurs almost exclusively in this region. And so here's another one. It's a protocadherin-like transcript that localizes. So this particular protocadherin is tremendously upregulated by symbiosis. And so we wanted to find out where it was, and lo and behold, it's this is GFP labeled, it's the Delta Lux genes of Vibrio fischeri, and this is the protocadherin, restricted to those places where bacteria do not reside. These protocadherins are proteins that mediate cell-cell adhesion in epithelia. So we were very intrigued by this, and we're following up on this. I'm missing something there. But the protocadherin transcript is restricted, like I said, to the ducts in this case. And so it's already known that protocadherins can be regulated by pathogens, for instance. So EPEC stimulates long-range microvillar dynamics, pulling protrusions towards the sites of bacterial attachment in a process mediated by protocadherin. So we think something like that is going on, and so we began to look at the protocadherins more carefully. It's a tremendously expanded gene family in the cephalopods, in squid and their relatives. So this really talented postdoc I have from Duke, Tara Essex-Byrne, looked, and there are 230 genes encoding protocadherin-like proteins. It's tremendously expanded in both our squid and the octopus. 129 of them are regulated in symbiosis, over twofold. It's huge. She's selected 8. Some are high, some are low, some are high in the aposymbiotic animal, non-symbiotic animal, some are high in the symbiotic animal. And so, I mean, it's just an amazing thing. And what her goal is, she's working with Scott Fraser, is to view all of this set of 8 protocadherin transcripts in time and space across the colonization of the host by Vibrio fischeri, Why all of these protocatecherons regulated? She's going to look at these 8 and see how they're deployed in time and space during colonization. So then in conclusion, I hope I've convinced you that specificity and symbiosis can involve, or likely does in almost every system, both biomechanical and biochemical factors. There's got to be a reason why in horizontally transmitted symbioses you pick up only a very small subset of the environment. And imaging, I think, is a very powerful teacher. And it informs us about both local and remote control of the association. So with that, I'm going to thank the people many of the people who have worked on this. This was the 25th anniversary of the study of Vibrio fischeri, and this is Ned Ruby, my partner, who runs the bacteriology side of this. And so this was a couple years ago in Madison before we moved to Hawaii, and this was this year's meeting. So we get together once a year, and we have what we call the powwow. There are now, I think it's 17 labs who work on the squid-vibrio system. And we get together so that we don't bump into one another, and that we have, that there's no reason in this system, which is very rich in asking questions, that there's no reason to compete, that's for sure. So we all get together once a year and talk to each other about what everybody's doing. But this would not have been possible at all. What we know about the squid-vibrio system wouldn't have been possible without all of the great people who have been willing to collaborate with us on this system. And so since 1989, when we started the system, we've worked with 33 labs at 23 institutions in 4 countries. And these are the heads of the labs that I've listed here. And we've really, really appreciated all of that collaboration. So with that, the star of the show is going to say good night. This is the best part. They bury in the sand every morning and then come out and forage at night. So I'd be happy to take any questions.
Do you want to watch it again?
You want to? Oh, sure.
Questions?
I can take questions. Well.
Thank you very much, Margaret, for wonderful, always new aspects. Coming back to the last part with your new in situ fish technology, it was not clear to me if, for example, these LPS binding proteins, are these induced by the symbiont, or does it also happen in the absence of the symbiont? So
the one that I showed you is extremely highly upregulated by the symbiosis.
It doesn't happen in the absence of the symbiont?
No.
It's not a specially regulated gene in the host?
No, it's not. No, it's not. So the symbiont, in an aposymbiotic animal, you don't see any signal.
So, Margaret, it's always a thrill to see how every little piece of the puzzle, every time I hear you talk, there's a new little piece. And I'm really, really intrigued by the strain diversity that you see and the co-colonization experiments. Have you had a chance, when you look at the difference between the dominant strain and what you're calling the social co-occurring strains, it's hard to imagine that they would have a chance under natural conditions. Do you know if when you go out and sample them in the wild, it's always the dominant strain that is indeed is the symbiont of the population?
It's definitely not. Because ES114, which is what all the genetics, all of the strains are from light organs. All of those strains that have been isolated were isolated from light organs. They're not environmental strains.
But if the dominant is so dominant, why isn't it dominant in the natural host?
The only thing that we can think of is that that in nature, in the field, that the social strain is better at being, that's what the culture work suggests. It's better at being a free-living organism and might dominate under those circumstances. The other thing that's really interesting is that
So you're saying, sorry, just to understand, you're saying it's more abundant in the environment.
We think so.
And therefore, it might have to take that because the dominant one is lower one.
Yeah, that's what we think. And we haven't completely proven that, but that's the going idea. Yes, I mean, that's the idea. But one of the other interesting things is I have a student working on the recognition and how long. So there's an interesting phenomenon. If the animal is colonized by ES114 persistently for 5 days, and you cure it, it will not recolonize with ES114. No, ES114 is a social strain. And it's amazing. We've done all of our work on something that doesn't make big aggregates. But so it speaks extremely well. So this social strain speaks extremely well to the host. So at 5 days, you cure and you cannot recolonize. The dominant strains that we've looked at, you can recolonize. And so, so there's something very interesting going on with the talking of a social strain to the host, and there might be a significant difference in how the host sees a dominant versus a social strain.
Margaret here. Wonderful talk. Great. So the other answer, of course, to the question to Nicole is that you have just different kinds of adaptations from the microbe to the host. But in that context, I still wonder, do you have a good answer? So if there are specific adaptations of the bacteria to the host, it requires that this information can really pass through many generations of the bacteria, including the generations that the bacteria has outside of the host, where it has no guarantee to meet the host again.
Yeah.
So do you have any idea how this can work?
Yeah, this particular system for the ecologists presents huge conundrum, because, they vent their symbionts every day at dawn, and there aren't that many squid out there. And it looks pretty much that the symbiosis is, sort of an ecological dead end. Many of the bacteria go into a viable non-culturable state, and the extent to which the dominant strain goes into a viable non-culturable state versus the other one is something we don't know yet. But
may there be another sort of advantage in sort of the free or the scent environment for these two phenotypes?
Yeah, I mean that's the idea that something about the social strain and something about the extra load of 250 kb that makes the social strain more fit in the environment.
2 more questions, Philip? And then you.
Thank you.
You may remember our discussion when we did the small study. The thing that puzzles me really is that you have 5 bacteria making contact with maybe 2 times as much host cells, and then you have a huge signal spreading.
Yeah.
Now, with all your beautiful visualization techniques, are you any further with your thoughts how the signal is actually spreading within the epithelium or in the host? I mean, are there any mechanisms?
Well, so what one has to believe is that there's something that Vibrio fischeri transports to receptors of the host cell that have this effect. And the most likely candidate, hands down, is the peptidoglycan monomer. And the reason is because in our system, as well as Drosophila, that peptidoglycan monomer has extremely high specific activity. And it's the exact, Neil Silverman tells me it's the exact same specific activity as in Drosophila. In other words, that molecule the animal just screams when it sees that molecule. And Vibrio fischeri exports that at levels that are the same as pathogens that export that. And so just constitutively. So we think that the animal is sensing that particular molecule. When we found out that there were only 5 cells, we knew that the bacteria had to be attaching to the cilia because there's such a huge flow field created by the cilia that we thought, bacterial molecules would be washed away. And in fact, when Melissa Altura in my lab she did a big project looking at attachment to cilia. Every bacterium that comes in is attached to a cilium.
Margaret?
Yes.
I was giving a lecture once describing your work,
Yeah.
which I like to do, as an example of a highly efficient, with great deal of fidelity of horizontal transfer from the parent to offspring. And an evolutionary biologist in the audience challenged me by saying, how do I know, or how do you know, that the offspring gets the vibrio from the parent rather than from someone else's parent in the sea?
Oh, you don't. So because the females, unlike other cephalopods that tend their young, the female lays the eggs and goes away. And 20 days later, the babies hatch into seawater that has a population of the squid. The farther you get away from adult populations, the harder it is for that water to colonize the animals. But there's a mixed population, mixed genetic population, and so it's not parent to offspring. Yeah, they pull it in from the seawater.
It seems we cannot let you go very easily, but now we have to because of the time.
Yeah,
so let's thank Margaret again.